Hi everyone, Varna here. Now let's continue with the mold biotechniques. Today let's see about the blotting techniques. What is blotting? Blotting, it is a technique for transferring DNA, RNA and proteins. So there are three types of blottings. That is, one is for DNA, another is for RNA and another one is for proteins. So in blotting we are transferring these materials, DNA, RNA and proteins, onto a carrier so they can be separated, and often follows the use of gel electrophoresis. The southern blot is used for transferring DNA, northern blot is for RNA and western blot is for proteins. So in blotting we are transferring the genetic materials like DNA, RNA and proteins onto a carrier, and here we uses gel electrophoresis for the separation of these genetic materials. Southern blot is for DNA transferring, northern blot is for RNA and western blot is for proteins. As I said, it is divided into three: southern, northern and western. Southern for DNA, northern for RNA and western for proteins. Let's see the history of southern blotting. Professor Sir Edwin Southern, professor of biochemistry and fellow of Trinity, developed a method in 1975, and this Sir won the Lasker Award for the Clinical Medical Research Prize for the method of finding specific DNA sequence. He developed this procedure at Edinburgh University more than 30 years ago. The technique is known as DNA transfer or southern blotting. This method involves separation, transfer and hybridization. So these are the steps. It is separation of DNA, then transferring onto the material and then hybridizing. They are routinely used in molecular biology for detection of a specific DNA sequence in DNA samples. The DNA detected can be a single gene or it can be a part of a large piece of DNA such as viral genome. Southern blotting combines agarose gel electrophoresis for the size separation of DNA with methods to transfer the size of separated DNA to a filter for probe hybridization. So first step is electrophoresis for the separation. Depending upon the size of the DNA, it is being separated by gel electrophoresis. In the next step, we are transferring or we are blotting to this sized DNA onto a filter membrane. So it is blotted or transferred to a filter membrane, and next step we are hybridizing it with the required DNA probes. The key of this method is hybridization. So as you know, what is hybridization? It is the process of forming double-stranded DNA molecule between a single-stranded DNA probe and a single-stranded target patient DNA. A probe is defined as a single-stranded piece of DNA which is labeled either with radioisotope or any other non-radioactive materials, and there is also fluorescent probes available, and non-radioactive probes are also available. So this probes will be having nucleotide sequence which is complementary to the target DNA, target patient DNA. The complementary strands will be available in the DNA probes which we are using. These DNA probes will either be radioisotopes or fluorescent probes. So depending upon, so after hybridization, depending upon the radioactivity or the fluorescence, we can identify how much amount that will be corresponding to the amount of hybridization, and thus we can find out the target DNA. So here the principle is going like this: we are using a DNA probe which is either radio labeled or fluorescent or non radio labeled, and we are allowing it to hybridize with the patient DNA. Patient target DNA is being hybridized. So the DNA probe will be having the complementary strands. DNA, the nucleotide sequence will be the complementary strands of that of the patient target DNA, and it is allowed to hybridize, and the excess will be removed by washing procedure. So, after that we are counting the, or we are calculating the amount of fluorescence or radioactivity. So, that amount, that will be corresponding to the amount of hybridization, and thus we can find out the target DNA. So, hybridization is the process of forming double stranded DNA between a single stranded DNA probe and a single stranded target DNA. So, these are allowed to hybridize, and we are calculating the amount of hybridization by the radio labeled or fluorescent labeled probes. Principle: the mixture of molecules is separated, the molecules are immobilized on a matrix, then the probe is added to the mixture to bind with the molecules. Any unbound probes will be removed. The place where the probes is connected corresponds to the location of immobilized target molecules. So, in the first procedure, the mixture of molecules is getting separated. The DNA is being separated. We are separating the DNA using gel electrophoresis. The DNA, depending upon their size, it is getting separated. So, the mixture is getting separated, and then we are immobilizing it onto a matrix. We are immobilizing this DNA, and the DNA probe mixture is added. DNA probe is having the complementary strand of the target DNA, and that is either radio labeled or fluorescent labeled, that will be added onto the immobilized matrix, and then the excess, and it is allowed to hybridize. After certain time of hybridization, it is washed away. So, after the process of washing, in which we are removing the unbound probes, next process is calculating the amount of hybridization. The amount of hybridization will give you the amount or the extent or the presence of the target DNA. So, that amount of hybridization is calculated by calculating the amount of fluorescence or amount of radioactivity, depending upon which type of probes which we are using. So, by calculating the radioactivity or by looking into the fluorescence, we can say the amount of hybridization, and that is giving you an idea about the target patient DNA. So, since the probes which we are using is having the complementary strands to that of the patient DNA, it will get attached to this patient DNA, and it will be immobilized onto that, and it will be shown during the reading time, and we can say that there is a presence of this target DNA, or we can also tell the extent or the quantity or how much amount is present, that can also be said. The place where the probe is connected corresponds to the location of the immobilized target molecule. Here it is the apparatus for southern blotting. This is the electrophoretic pattern. Electrophoresis is done in order to separate the DNA depending upon their sizes. Charges will be supplied on either sides. You can see the chambers. Charges will be applied on either sides, and the DNA will be separated depending upon their sizes. In the next step, after separation depending upon their sizes, it is blotted onto a filter paper. This is the paper which we are using, Wattman 3mm paper, in which they are blotted. Here you can see the whole apparatus, the down portion. Here it is the electrophoresis for the separation of DNA depending on the size, and here the filter paper is kept for the blotting where it is being transferred, and here this bottle is kept for the weight purpose, and the nitrocellulose membrane also you can see, onto which it is blotted. Okay. It is the blotting apparatus where after the electrophoresis the gel is getting attached to the membrane where it is getting blotted or transferred. Then filter paper is being kept, and on the top portion there is weight kept in order to place the apparatus on proper position. Let's see in detail the steps of southern blotting. In the first step, it is treated with restriction enzyme, that is in order to digest the DNA with an appropriate restriction enzyme. It is getting fragmented. After the application of restriction enzyme, this DNA is cut into smaller portions, and we are getting fragments of DNA molecules. The complex mixture of fragments is subjected to gel electrophoresis in order to separate the fragments according to their size. In the next step, after fragmentation, we are allowing it for migration through gel electrophoresis so that they are getting separated according to their size. According to their size, they are getting separated. In the next step, the restriction fragments is present in the gel are denatured with alkali, and it is transferred onto a nitrocellulose filter or nylon membrane by blotting. So after separation by electrophoresis, it is denatured. The DNA is denatured by the addition of alkali, and it is blotted onto a nitrocellulose filter or a nylon membrane. So during the blotting process, the DNA is getting transferred onto that membrane. This procedure preserves the distribution of the fragments in gel, creating a replica of the gel onto the filter. So we are getting an exact replica of the gel electrophoresis onto a filter. Next step, this filter is incubated under hybridization condition with a specific radio-labeled DNA probe. So in the next step, we are taking a DNA probe which is complementary to the target DNA, that is either radio-labeled or fluorescent labeled. So this radio-labeled DNA probe is added onto the filter. The filter, it is getting incubated with the DNA probes, and it is allowed for hybridization. So after certain time of hybridization, it is removed. The probe hybridized to the complementary DNA restriction fragment. And in the next step is washing, that is excess probe is washed away and the probe bound to the filter is detected by autoradiography, which reveals the DNA fragment to which the probe is hybridized. So after washing, we are reading the extent of radioactivity by autoradiography, and that is revealing the DNA fragments which is hybridized. Let's see the procedure once more. First, initially the DNA is being separated, and then it is fragmented using restriction enzyme. Then next step, it is separated. The DNA fragments are separated depending upon their size by applying gel electrophoresis. Then this electrophoretic gel pattern we are getting, gel pattern of separated DNA. This electrophoretic gel pattern is then blotted or transferred onto a membrane. Into the membrane it is being transferred. So we are getting just a replica of that, and then this membrane is allowed to hybridized with the DNA probes, which is either radio labeled or fluorescent. Here we are using radio labeled probes. So this membrane is having just the replica of the electrophoretic pattern, and that is incubated with the DNA probes, which is radio labeled. So the DNA probes will be having the complementary strands, so it will be getting attached. It will be getting attached onto the target cells, and wherever there is attachment or a hybridization, we can say that there is hybridization occurring, and we can find out the extent or the amount of target DNA, and that is the excess will be washed away. Excess unbound DNA probes will be washed away, and we are reading the radioactivity. This is the same thing. First procedure, that is electrophoresis, unlabeled DNA, that is the patient DNA, which is cut by using restriction and nuclease, and then it is electrophoresis. Next procedure is electrophoresis, and the labeled DNA, depending upon their sizes, they are getting separated, and this separated agarose gel is blotted onto a membrane. Here you can see the nitrocellulose membrane. The nitrocellulose membrane is kept on top of the agarose gel, and a sack of paper is kept on top of that. That is Wattsman paper. 3 mm paper is kept on top. Nitrocellulose paper or nitrocellulose membrane, it is getting blotted with the gel. Whatever pattern is on the gel, DNA pattern is on the gel. Electrophoresis, after running the electrophoresis, whatever pattern is there, that is getting blotted or transferred onto that membrane. So we are getting just a replica of that. Then we are removing the nitrocellulose paper, and it is getting incubated with the DNA probes. DNA probes having complementary strands, so they are getting hybridized. After a certain period of hybridization, it is washed away. The excess is washed away, and then we are reading the amount of hybridization to find out the target DNAs. Next, we will see the applications of Southern blotting. Southern blot are used in gene discovery, gene mapping, evolution and developmental studies, diagnosis and forensic. These blotting techniques is used in DNA fingerprinting, preparation of RFLP maps, and identification of transferred genes in transgenic individuals, etc. Southern blot allows investigators to determine the molecular weight of a restriction fragment and to measure the relative amount of different samples. Then it is also used to detect the presence of particular bit of DNA in a sample and analyze the genetic pattern which appears in a person's DNA. All these studies is possible with Southern blot, including the DNA fingerprinting. You all know what is DNA fingerprinting and how it is used in forensic, how important it is. Okay, next we will move on to Northern blotting. Northern blotting is just techniques for the detection of specific RNA. Instead of DNA, here we are taking RNA sequences. So Northern blotting was developed by James Alvin and George Stark at Stanford University and was named as such by analog of Southern blotting. Next we will see the principle is same as that of Southern blotting, and let's see the steps. First, the RNA is isolated from several biological samples like tissues and various developmental stages of some tissues, and this RNA is more susceptible to the degeneration than DNA. The next step, the samples are loaded onto a gel and the RNA samples are separated according to their size on the agarose gel. So first one is isolation, next one is loading onto the gel electrophoresis and it is getting separated according to their size. Then next, the resulting gel following after the electrophoretic run. After the electrophoresis, the gel is then blotted onto a nylon membrane or a nitrocellulose filter paper by creating the sandwich arrangement. So next step is same as that of the other, that is it is placed onto the electrophoretic pattern and we are blotting or transferring the RNA onto the membrane and we are getting the replica. The membrane is placed in a dish containing hybridization buffer which is having labeled probes. So in the next step, it is allowed for hybridization by radio labeled probes. Thus, it will hybridize to the RNA on the blot that corresponds to the sequence of interest. So, here the RNA probes, which is radio labeled, which is having corresponding strands as that of the target RNA, that is getting hybridized. And we are next step, we are washing the membrane in order to remove the unbound RNAs. That is removed. Then in the next step, the labeled probe is detected via autoradiography or via chemiluminescence reactions. If a chemically labeled probe is used, we are using chemiluminescence, then fluorescence, autoradiography. All those methods are being used in order to read or detect the extent of hybridization. In both cases, this results in the formation of the dark band on the X-ray films, and now the expression patterns is of a sequencer obtained. So, by reading we can get the amount of hybridization. All these are wherever there is the labeled DNA probes, we are getting dark bands on the X-ray films. So, we can detect the presence and we can say the extent of presence of these target DNAs. Moving on to its application. Northern blot is used for the study of gene expression at the level of mRNA, messenger RNA, then detection of mRNA transcript size, then RNA degeneration study, then RNA splicing, RNA half-life, all these studies it is being used. Then it is often used to confirm and check the transgenic knockout mice. All these studies it is being used. Disadvantages of northern blotting, that is the standard northern blot method is relatively less sensitive than nucleus protection assay. Then detection with multiple probes is not possible. Then if RNA samples are even slightly degenerated by the RNAs, the quality of the data and the quantitation of expression is quite negatively affected. So the degeneration process is highly sensitive because RNA is more prone to degeneration. Next method, that is Western blotting. Western blotting is an immunoblotting techniques which rely on the specificity of binding between a protein of interest and a probe. So here we are using a protein. We are identifying the presence of a specific protein. So it is depending upon the specificity of that protein and a probe. Probe in the sense antibodies. Antibody raised against the particular protein. And these antibodies are allowed to, the labeled antibodies are allowed to bind with the protein. And we are calculating the extent of binding. So they are allowed to allow the detection of protein of interest in a mixture of many other similar molecules. Let's see the steps. First step, the protein samples is subjected to electrophoresis on an SDS polyacrylamide gel. So it is as usual, it is running electrophoresis in the first step. Then next is electroblotting. It is bloated or it is transferred. The separated proteins is transferred from gel to the surface of nitrocellulose membrane. Then this blotted paper is incubated with the generic protein such as a milk proteins or BSA, which binds to any remaining sticky places onto the nitrocellulose. And then an antibody that is specific for the protein of interest is added to the nitrocellulose sheet and it reacts with the antibody. Only the band containing the protein of interest binds to the antibody, forming a layer of antibody molecules. So antigen antibody binding is taking place when they are incubated with generic proteins. So after that it is washed. After the washing process, it is removing the non-specific bound antibodies 1. So the first added antibodies excess which is not been bounded will be removed from it. And next step is that we are adding a second antibody. Antibody 2 is added, and it is specifically recognizing the Fc domain of the primary antibody and is binding to it. And this antibody 2 will be radioactively labeled, and or it can be covalently linked to a reporter enzyme. So it is the antibody 2 is binding to antibody 1, which we added initially. So they are linking together, and this radio labeled antibody helps in the visualization of the protein antibody 1 antibody 2 complex. So first we are adding with antibody 1 to the proteins, and they are binding and forming a complex. The excess is washed away, and secondly we are adding another antibody. Antibody 2 is added, which is radioactive or radioactively labeled or covalently linked to a specific enzyme. So this antibody 2 is making a complex with antibody 1, and altogether we are getting a protein antibody 1 antibody 2 complex. So since antibody 2 is radioactive, it can be read or recognized, making it possible to get an idea about the proteins. Applications: it helps in the confirmatory HIV test. Western blot is also used as definitive test for BSC, bovine spongiform encephalopathy. Some forms of Lyme's disease testing is also done with Western blotting. Thank you.